While we focused during the first lesson on terminology and archaeobotanical research questions, we will continue in this lesson with the more practical issues of archaeobotany. Here you can see the archaeobotanical workflow of the data collection process and possible disturbing factors. We already discussed taphonomic processes. Starting with the excavation, there are various issues that may lead to limitations in the acquisition of samples. This is often related to the type of excavation, for example, if the site is a rescue excavation or the financial situation does not allow large-scale excavation. Even if this is not the case, the number and volumes of samples taken from the site may be too small to allow representative data acquisition. During sample processing, the methods chosen or unsuitable sieve sizes may result in considerable loss of material. While sorting archaeobotanical samples, insufficient knowledge may also lead to selective representation of remains. The same applies to morphological identification, which may also suffer from bad preservation of the remains. And here the steps of the data evaluation. They include the classification of data, for example, ecological grouping of wild plant taxa, followed by numerical data analysis, which in archaeobotany is only rarely realized by applying multivariate statistics. The results are interpreted in relation with the questions on the ancient environment and economy. However, the more incomplete the data list is due to the just mentioned possible limitations, the lower the reliability of the interpretation will be. At the beginning of each archaeobotanical study, we need to develop a site-specific sampling strategy. The way of sampling determines the kind of conclusions we can draw from our data. Very often, a combination of different sampling methods is reasonable, because each sampling method has advantages and disadvantages. Generally, samples should derive from undisturbed and well-dated or datable contexts. As many samples as possible should be taken. In general, there are two main sampling strategies that are applied in archaeological sites: judgmental or purposive sampling and systematic sampling, which are ideally applied in combination. Judgmental or purposive sampling is mostly based on empirical knowledge, that is, dark sediments often contain carbonized plant remains, and they are chosen for sampling if seeds can be seen by eye. This kind of sampling is most frequently applied, but additional sampling of less conspicuous contexts is necessary to receive a representative assemblage. Systematic sampling is conducted along a predefined grid, so that samples are taken over the whole excavation area in the same inter-sample distance. This kind of sampling and the following analysis are possible in large-scale research projects with considerable funding of archaeobotanical research. Within settlement sites, such as Near Eastern tell sites, the goal is to sample large volumina by gathering all sediment within a sector of the grid. The amount of samples and sediment volumes required for generating a large dataset are generally larger than in waterlogged sites because the find density is much lower in non-waterlogged sites. In waterlogged sites, archaeobotanical samples are usually taken as sediment cores. The black Dots on the picture to the left on the plan of the Neolithic lakeside settlement of Arbon-Bleiche indicate the coring locations. At the beginning of the investigation, almost no structures and also no layout of houses was visible, and became only recognizable after the systematic analysis of the archaeobotanical samples. An additional benefit of systematic sampling is the possibility to investigate activity zones, as has been achieved at the Neolithic site of Sipplingen. Activity can be in relation with food processing, and its recognition may thus determine specific functionalities of areas or installations. The localization of activity zones of plant use is an important aspect for recognizing the economic and social organization of households. Spatial patterns may even allow conclusions about cognitive abilities. Sampling with cores or in profiles may also be conducted in lakeside settlements that are currently not underwater, because the sediments are relatively soft. We already saw sampling of sediments with plastic tubes. Here another example from the final Neolithic settlement of Torwiesen in southwestern Germany. Profile columns using flower boxes is an alternative if the sediment is unsuited for hammering down plastic tubes. This, however, rarely allows true systematic sampling because profiles are usually not present in an exact grid over the excavation area. The example shows the light lake sediments and dark cultural layers from Neolithic Horgenscheller. For complex stratigraphic units, profile columns are always to be preferred. Having seen the most frequent sampling strategies of judgmental and systematic sampling, we will shortly have a look on a less frequent method, that is, random sampling, which we see on the left in comparison with systematic sampling on the right. Random or probabilistic sampling is based on the goal to sample each excavation unit with the same probability. To achieve this goal, the excavator must have an overview on a predefined excavation area. That is, the contexts and structures need to be clearly visible, and the stratigraphic level must be identical in all excavation areas. After mapping of the first excavation level, the contexts are grouped and a decision on the size of the subsample will be made. Contexts will be numbered, larger structures are geometrically subdivided, and their sections will also be numbered. By using a random number table, as you see here in the right, the amount of numbers needed to subsample the excavation level will be blindly chosen. There are some disadvantages in using this method. First, in most excavations, the preconditions for random sampling cannot be fulfilled. If too few samples are chosen, representativeness will be lost. Contexts with well-visible remains may not be sampled, which will result in a loss of information. Therefore, random sampling should only be used as an additional method. In the left picture, you see an example for a combined random and judgmental sampling. There were only post holes visible. Roughly 30% of the post holes were random sampled. Additional judgmental samples were taken to consider also contexts that contained plant remains visible by eye. To avoid gaps, further samples may be taken. As we were looking at the sampling strategies, we already made acquaintance with different kinds of contexts. We will continue now with considering the most frequent context types in archaeological records. Here you see two examples for cultural layers: a burned layer from Middle Bronze Age Troy and floors within the house structures of Middle Bronze Age Tel Musan in Syria. The term cultural layer is very broadly used by archaeologists to describe different context types, such as floors or burned layers in settlement sites. They contain intentionally or unconsciously deposited sediments by ancient humans. Only when mechanical influence is high, as for example on floors, the fine density will be low. Ovens and hearths are relatively frequent in settlement sites. They, however, do not always contain a high amount of plant remains, because they have been regularly cleaned during the time of their usage. In the case of the Middle Bronze Age cooking area at Troy, a settlement fire led to the preservation of the life situation, that is, a cooking area at the moment of its usage. Therefore, a large number of well-preserved plant remains were found in pots next to the oven. Pits are in most cases the result of active disposal of waste. Their original function may differ and not have been related to waste deposition. In some cases, they may have been used as storage pits, and only later, when storage was no longer possible, garbage was disposed in them. Roman and medieval wells have also often been used as waste disposals during later settlement phases. As wells reach to the groundwater level, preservation of organic material is good in the lower part of the well. However, the findings in the well derive to more than 90% from secondary filling after the primary use. Most fillings of ditches are of secondary nature, that is, they are younger than the period of use of the ditch. Ditches are only suitable for archaeobotanical investigation when a stratigraphic sequence is clear and datable. The samples need to be taken at several locations of the ditch to get a representative result of the deposits. Other more rare contexts are cesspits and latrines. If the water table did not considerably change throughout time, they can be very rich in uncarbonized plant remains and provide a nice information basis on what people were eating in the past. While tombs and graves are interesting for cultural aspects, they suffer from limited preservation chances for archaeobotanical remains, and often contain plant remains only when they have been carbonized through ritual burning. This list shows some standards for the number and volume of archaeobotanical samples from archaeological sites. Waterlogged contexts, such as in Neolithic lakeside settlements, which usually have a high fine density, so that is more than 1,000 remains per liter of sediment, should be sampled by taking a minimum of 8 samples per context, as for example a house, for representatively covering the typical assemblage from such a context. The sample volumes should be between 5 to 10 liters, but will then be subsampled by taking 1 to 2 liters before wet sieving. Dry contexts usually contain much fewer plant remains, so less than 10 seeds per liter of sediment. Sample volumes should therefore be larger, and typical standards are 10 to 30 liters of sediment. If dry contexts have high fine densities, as for example a burned storage facility, smaller volumes can be taken, but if the context is extensive in size, more samples covering different areas need to be taken. Archaeobotanists often use the term representative. But what is meant by representativeness? In general, the goal is to work with samples that are large enough to guarantee that all taxa that have been present at the sampling location are covered. That is, each plant remain should have the same chance to be recovered. This can only be achieved if all contexts are sampled equivalently or if a surface has been sampled systematically. Let's have a look on the sample processing. A pre-treatment of the samples is usually only required when sediments are very loamy and closely stick to archaeobotanical remains. Some pre-treatment methods are described in the textbook by Pearsall, published in 2000. To loosen the sediment attached to carbonized remains, drying in a cabinet desiccator at about 30 degrees centigrade for a minimum of 48 hours, followed by soaking in water may help. Another method would be to put the samples in a refrigerator for a minimum of 24 hours, followed by slow defrosting and careful flotation. Some researchers also use a solution of a detergent such as washing powder to soak the sediment before wet sieving or flotation. Standard mesh sizes to recover all remains are a set of sieves with 42 mm, 1 mm, 0.3 mm down to 0.2 mm. As concerns the processing methods, a couple of methods are used, such as washover or wet sieving. You can find a good overview on different sample processing methods in the textbook by Jacomet and Kreutz. So what can be found in the different mesh sizes? Starting with the 4 to 2 mm sieve, we will find seeds of crop species, that is larger fruits, larger seed remains, and also identifiable charcoal pieces. There will be bones of small mammals and fish, mollusks, and even small artifacts in these sieves. The 1 mm sieve will mostly contain chaff remains of crop species if agriculture was practiced on the site, and then larger wild plant seeds. Plant fragments and fragments here of mostly cereals, and these are sometimes hard to be identified. Then we may find epidermis fragments, again mollusks, ostracoda, which are small crustacea, whelves, and then we may also find insect remains in these 1 mm sieve sizes. Then the smallest, that is 0.35 mm or even smaller, will contain mostly small weed species, small wild plant seeds. Oogonia of the Karatsay can be there if the site was next to a water source. And then we may also find again ostracoda and even parasite eggs. The most frequent sample processing method in arid to semi-arid environments with loose to dusty sediments is flotation. The principle works through the relatively light carbonized plant objects that easily float to the surface when poured into water. The method is most efficient because it is the least destructive sample processing method and allows extraction of large amounts of carbonized material from large quantities of sediment. Early applications were conducted by hand, that is a so-called bucket flotation, and today most excavations use machine flotation. The remnants that stay in the mesh in the largest barrel are the heavy residues, and they always need to be sorted or at least checked for unfloatable remains. Here just another example of the flotation machine at the excavations of Sinchili. After sample processing on site, the samples are usually transported into an archaeobotanical laboratory where they are further processed and sorted. In the lab, the extract or flot is generally sieved again to split the sample into different fractions and to ease the sorting process. The sorting process is conducted with a binocular under 5 to 30 times of magnification. Seeds and chaff remains can be already grouped by using morphological criteria. Identification of seeds and chaff is conducted with identification keys and a comparative collection of modern seeds. For this step in data acquisition, comprehensive botanical and morphological knowledge is necessary. In case of large find numbers, it may be required to subsample in order to manage the workload in relation to time and available financial means. Particularly the small fraction contains large amounts of inorganic remains and unidentifiable plant fragments, while only few small wild plant taxa are preserved. To receive representative results, a standardized subsampling must be used, which is possible with a riffle-type sample splitter applied to dry sediments or dry flotation fractions. To correctly apply the method, the minimum size of subsamples necessary to produce representative results must be calculated first. For this process, the sample or fraction has to be divided into 8 subsamples by using the sample splitter. Each subsample has to be analyzed separately. If they all contain the same species and more or less the same numbers of records, 1/8 for a subsample would be sufficient, and this size could be applied to other samples of the same site. If there are subsamples that do not contain all botanical taxa, 1/4 of a subsample would be more appropriate. In most cases, subsamples of 1/4 are sufficient for the smallest fraction. For waterlogged samples, other methods must be used. After sieving, the volumes of the different fractions need to be measured. Subsamples are taken with a spoon from different places within a sieve. It is, however, more difficult in this case to get equally sized subsamples. The counts of records in subsamples will be projected to the complete sample. You can find additional information on subsampling of waterlogged samples in the textbook by Jacomet and Kreutz. The identified plant remains are counted and noted in a taxa list. Charcoal is often weighed. Counting follows certain standards. It is important to treat different categories of the same taxon separately. For example, grain and chaff need to be counted separately because they represent different aspects in subsistence and agriculture. Grains may derive from different activities, but are generally the crop processing byproduct, intended to be stored and consumed, while chaff remains are crop processing byproducts and have entered the archaeological record as waste. Seed fragments that represent at least 1/4 of the seed are added up to complete seeds. That is, 4 fragments of a grain that represent 1/4 each are counted as 1 grain. Other ways to count fragmented material is weighing, which is particularly practiced with wood charcoal, which is generally sensitive to post-depositional fragmentation. Although they break in the same manner and the mass per fragment of each sample is proportional to the fragment count, Comparison between sites are impossible if fragment counts are used. Therefore, weighing should be preferred for charcoal pieces. Other basic laboratory analyses are morphometric studies that have been earlier used to investigate shape variations of cultivars in prehistoric and historic sites, such as for grape. Since digitalization of measurements through application of morphometric software has entered the archaeobiological laboratories, more complex shape variation could be investigated and thus allowing evolutionary conclusions based on the investigation of large amounts of material. They can also be helpful in differentiating different wild plant species, which are otherwise difficult to be morphologically grouped. Recently, measurements of wild progenitors of modern crops have been used to investigate the domestication rates in cereal species. The basic step in data evaluation is grouping according to specific criteria. We already learned in the first lecture that plants show qualitative differences in the usefulness for humans and in their ecology, and can be grouped according to such criteria. Such groups help to structure and thus to understand large and complex datasets. Starting with subsistence-based grouping, there are a number of options how plants can be used. In categorizing the identified species according to these usage groups, conclusions on ancient diet, crop plant history, handcrafts, and trade are possible. The archaeological context is of basic importance and obligatory for a reasonable archaeobotanical interpretation. Here an example from the early Bronze Age palace area at Khirbet es-Serakon in Jordan. The archaeobotanical assemblages indicate spatial patterns that are directly related to the contextual situation. A distinct difference is visible between the samples in the altar area with large amounts of small-seeded pulses and emmerchaff, in contrast to the botanical assemblages outside this area. These differences led, together with the faunal analysis, to the interpretation of the presence of gut contents from animal offerings, because small-seeded pulses and chaff remains are typical for animal feeding. Ecological grouping allows conclusions on ancient vegetation and habitats that have been preferred by ancient humans. By relying on uniformitarian principles, modern ecological characteristics are applied to archaeobotanical assemblages. This has to be done cautiously because modern species may have changed their ecological habitats. If we are considering different phases or periods of a location, environmental change may be observable. Depending on the detail of information, anthropogenic influence may be visible in the diachronic comparison of the archaeobotanical assemblages. As we have seen in the first lecture, there are a number of tools that allow structuring of our archaeobotanical data into ecological groups. These tools either derive from the scientific fields of geobotany or from phytosociology. While geobotany is concerned with the geographic distribution of plant species, that helps the archaeobotanist to understand the environmental factors that explain why a species is found in a site, phytosociology is a key to understand why different plant species occur together and is also based on ecological requirements of plant species. There are different approaches to categorize archaeobotanical data. The indicator values by Ellenberg have been occasionally used by archaeobotanists, but they are very specific and have been developed for the Central European flora. The method uses several ecological characteristics for each species. Each of the ecological factors is arranged into values between 1 to 9. Using indicator values allows a direct quantitative approach, but is also restricted to records which have been previously identified to the species level. Unfortunately, as a matter of preservation and intergeneric morphological diversification, only few archaeobotanical taxa can be identified to the species level. Therefore, the use of Ellenberg indicator values is only recommended for broad archaeobotanical assemblages with many identified objects to the species level. More common and also more practicable is a grouping into broader categories, as for example suggested by Jacomet. Here, the archaeobotanical taxa are attributed to habitats based on phytosociological units. Such systems can be kept quite basically, in case the details are unknown. That is, if not many seeds could be identified to the species level. The most basic and in many cases most correct generalization would be a grouping into categories such as water plants, alluvial vegetation, open habitats, and woodland. Here an example for ecological grouping of the archaeobotanical assemblage from Abu Huraira. In this case, floristic information has been used, that is, the availability of plants and seeds throughout the months of the year, which for the seeds corresponds to the fruiting time. The investigation of the plant remains according to this information showed that the settlement was occupied year-round. Let's consider now some basic quantitative methods. Differences in seed production, as well as differences in seed counts in individual samples, leads to differently represented plant remains. But how can we compare different samples with different total seed counts? A barley ear has about 30 grains, a poppy capsule more than 1,000 seeds. How do these individuals compare? One way to avoid differently represented taxa is by using presence-absence data as shown in the table. Presence-absence data is particularly powerful if used in a supra-regional approach, as in the example here, where the occurrence of domesticated species in the Fertile Crescent has been evaluated. The darker shaded cells indicate secure identification, while the lighter shaded mark the less secure identification of a taxon. Overall, the analysis of presence-absence of domesticated species and their wild progenitors indicates that a regular presence of domesticated plants, detectable on the basis of their morphology, is noticed in the pre-pottery Neolithic B period from about 8,500 calibrated BC onwards. Switching back to our previous sample table, we recognize the relation of presence-absence data to the ubiquity of frequency, which is also a common archaeobotanical quantification measure. The ubiquity is the presence of a taxon in the whole set of samples expressed in %. Ubiquity is often considered to inform on the importance of a species because if the ubiquity is high, the species appears in many samples. The problem with this kind of argument is that ubiquity is only a reliable measure when the number of samples is high, that is 10 samples as a minimum, and they have to be evenly distributed over the excavation area. The ubiquity of taxon X on the left would be 40%, on the right only 30%. The information quality, however, strongly differs and needs to be taken into account in the interpretation. Comparisons between individual samples are only possible if they are similar in size. The reason for low find numbers may be originally few remains that have been deposited, bad preservation, or small sediment samples. To exclude biases through small sediment volumes, there are standard sizes for archaeobotanical samples. However, during excavation, it is not always possible to sample equally sized sediment volumes. Therefore, the measure of find density is applied by calculating the number of finds per liter sediment. This requires a thorough documentation of the sediment volumes floated on site. Before the samples are compared, the find density of each taxon would be calculated, for example, as a rule of 3. Using the data visualization options of a spreadsheet software like Excel, there is no need to calculate find density when comparing sample composition, for example, in bar chart diagrams, if percentage proportions of the taxa are shown. Percentage proportion is easy to calculate on the basis of the total of records in one sample. Here an example how percentage proportion is used for demonstrating the changes in woodland composition over a stratigraphic sequence from the Neolithic to the Chalcolithic. Particular with small datasets, this kind of simple numerical data evaluation is sufficient because samples are directly comparable. However, one needs to be careful about the number of finds in a sample, because samples with few finds can be misinterpreted for their individual taxa, because these can appear to be numerous, although they are only represented with few remains. On the right, an example how percentage proportion can be used to investigate crop processing stages. The aim of growing a crop is to obtain a clean product that may be further processed into food. Information about how crops were processed and stored comes from both wheat seeds and remains of the cultivated plant itself. After the harvest of cereals, for example, the farmer is left with a large amount of undesirable wheat seeds, chaff, and straw. Ethnographic work has demonstrated that there are relatively few ways in which processing sequences can be carried out. You can see crop processing stages for wheat and barley in the left picture. Archaeobotany can assign chart assemblages to a single processing stage. The figure on the right provides the results of sample composition from an archaeobotanical analysis, visible in A, next to those from an ethnographic study, visible in B, and may allow discussion about which processing stage may be represented in the archaeobotanical record. For larger datasets, multivariate statistics may be necessary to recognize patterns in the data, for example, to address questions of diachronic or supraregional development of archaeobotanical assemblages. Multivariate describes the principle that in such kind of analysis many variables can be introduced. Dealing with large amounts of data often involves a high diversity in the dataset, which complicates the recognition of patterns. Samples with low seed finds and taxa with low ubiquity can bias the analysis and appear in the graphical output as noise. It is therefore required to modify the data and ensure that the samples are comparable. As we already learned, number and size of samples as well as archaeological contexts determine the representativeness of archaeobotanical samples. As these factors are no longer visible in the statistical output, they need to be unified before data analysis. This is called data compression and reduction, or data manipulation. The first step in data manipulation is to check whether two or more samples derive from the same archaeological excavation unit and represent the same structure that has been sampled several times. In case two or more samples are from the same unit, the record numbers of each taxon have to be added up into one sample. Through this process, the total number of records and taxa is increased, which leads to overall fewer but larger samples. If samples are too small, that is, they contain less than 50 to 100 remains, they have to be excluded from the analysis because they tend to create atypical patterns. If the ubiquity of taxa is below 10 to 5%, they are usually eliminated to avoid outliers by rare taxa. Because we do not wish to lose these data, we try to keep the rare taxa through adding them into equal categories on the next higher level of identification. Through this process, the ubiquity of the taxa category increases. This kind of data compression keeps a maximum of data in the data table. After data compression, the loss of data through reduction is generally not more than 2%. If no compression has been conducted, the loss of patterns can be considerable. It is also important to know that the step of data manipulation may have a larger effect on the final result than the statistical method applied. Therefore, a number of archaeobotanists disclaim multivariate statistics with the argument that statistics would falsify the results. Fact is that without data manipulation, the unevenness of the data is reflected in the output, but uneven data is often the result of an inappropriate sampling strategy. This means without data manipulation, we get a picture of the methodological problems during the sampling process rather than a representative pattern of the composition of the plant assemblage. Exception are small samples which may contain taxa that are not represented in the larger samples. By eliminating these small samples, there would indeed be a loss of information. Therefore, statistics are used as an additional method alongside basic numerical methods which can be conducted in a spreadsheet software. In general, archaeobotanical data are relatively uneven, or in other words, do not follow a Gaussian distribution curve. That is, there are large differences in numerical counts which may lead to distorted patterns through overproportionally represented taxa. The importance of a normal distribution counts particularly when tests of significance are conducted. Therefore, data transformation is conducted for example through a logarithmic calculus or by raising the records to a power of 2. There are different multivariate methods in use. However, only a small number has been established for archaeobotanical material. Correspondence analysis is amongst those most widely used in archaeobotany. Correspondence analysis is also frequently applied in ecology as well as in archaeology. Pattern searching with correspondence analysis aims to produce a picture of the relation between samples and taxa. The simplified two-dimensional figures show the originally multidimensional data pattern. The variables, that is, the taxa with their different counts, are plotted along the axis, which show the degree in variation. The x-axis shows the highest degree of variation. In the diagram to the right, the samples from Troy are most different from all other samples. Testing classification of extrinsic variables, a reasoning for the taxa variation can be developed. Such classifications may be either sample or taxa-oriented and can be chronological, ecological, or contextual in nature. In general, not only one classification applies, but combinations of factors that overlap more or less. Here a simplified example demonstrating how the raw data in the table is represented in a correspondence plot. Again, the principle behind the diagrams is that they represent simplified two-dimensional figures of an originally multidimensional quality of the data clustering. The ordination technique arranges the samples along the axis according to the spectra and number of taxa. So, the position of each sample is shown relative to all other samples and to all the species, and of each species relative to all other species and to all the samples in the analysis. In correspondence analysis, the first principal axis, which is the horizontal axis, accounts for the largest proportion of variation, that is, has the highest eigenvalues among the data. Only to shortly mention, the naught-naught coordinate is also very important for the interpretation, as the vicinity of data points to the naught-naught coordinate indicates similarity in composition for samples and high ubiquity of the particular taxa. To simplify, distance is the visualization of difference, vicinity of data points stands in most cases for similarity or correlation. It is assumed that the produced axes represent the underlying causes of taxa variation among samples. As such, the diagrams are used to build hypotheses on the causes. Hypotheses are tested via classification of data points according to extrinsic variables, such as sample chronology and origin or taxa ecology. Here a dataset from different sites in the Near East after elimination of crops and probable weeds. Interesting is the clustering of sites according to their geographic position, indicating that wild plant assemblages reflect geographic patterns rather than having a chronological attribution. Further categorizing of the sample data shows that precipitation plays a role in clustering of the taxa. Even outliers, such as the position of the Syrian site Ain Dara close to the Aegean sites in the previous diagram, become explainable. Mean annual precipitation at Ain Dara is higher than at other Syrian sites. The wild plant spectrum of Eindara is therefore more similar to those of Aegean sites, whereas no such patterns were visible in the analysis of the crop assemblages.